Okay, first of all, I'd like to thank Linda and Roger for having invited me, having most importantly organized this meeting that I think is very useful to the field, and yes, having invited me. So what I'm going to present today is completely preliminary data. Some of them I have not even seen myself. It is more a data club and a lab meeting. There are many holes in the stories, and I will be the first one to acknowledge it. So if you show them to me, I really know that they are there already. So the question we are asking, and it is in the context of this meeting, which is between bone and skeletal muscle interaction, this concept of bone and skeletal muscle interaction implies that there are molecules going back and forth or one way only between bone and muscle or muscle and bone. And the question we are asking is bone regulating any aspect of muscle biology going from mass to function? And the underlying hypothesis is that if this meeting has a reason to exist, and it does, there must be this interaction between bone and muscles, or muscle and bone, must rely in part on specific molecules, bone-specific or muscle-specific molecules that speak to the other organ. Because if these molecules are not cell-specific, then the interaction between bone and muscle is only part of a general interaction between every organ and every other organ. So, with that in mind, this question, we addressed it from the bone aspect, and I think it is now possible to address this question because in the last 5 or 7 years, bone has become an endocrine tissue making 2 hormones. I'm going to speak of only one, which is osteocalcin. It's the undercarboxylated form of osteocalcin is secreted, is an osteoblast-derived hormone that has many good functions. It increases beta cell proliferation, it increases insulin expression and secretion, insulin sensitivity, and energy expenditure. So as a whole, it favors glucose homeostasis and promotes energy metabolism. On the top of it, and in a totally unrelated manner, osteocalcin favors male fertility by increasing testosterone synthesis. The only known receptor for osteocalcin is a GPCR called GPRC6A, and we know from genetics that GPRC6A is the only receptor, at least in the Leydig cells of the testes. That does not mean that it is the only receptor in other organs, but at least in the testes it is. The originality of bone as an endocrine organ and osteoblast as an endocrine cell is that it contains another gene, ESP, that has an endocrine function, and yet ESP is not a secreted molecule. It's a tyrosine phosphatase, which, through a complicated mechanism I will not present today, inhibits all the function of osteocalcin. So in a cartoon, and taking glucose metabolism as a tool, osteocalcin, when it is uncarboxylated, is a hormone that acts on beta cell to increase proliferation and insulin production, and on muscle, liver, and fat to increase insulin sensitivity. And the result is to improve glucose handling. Behind the scene, behind these glamorous molecules, there is ESP, whose function indirectly is to inhibit osteocalcin by carboxylating it. And as a result, all the functions of osteocalcin are inhibited. And the interplay between these 2 molecules in the osteoblast explains the homeostasis function of the osteoblast. As a result, also, we have 2 models to study osteocalcin. One is a loss of function model, the classical knockout, osteocalcin knockout. And one is a gain of function model, ESP knockout. And this is very fortunate because every result of any experiment done with a knockout of osteocalcin can be verified with an internal control that is built in the system, which is the ESP knockout. And I'm going to show an example of that before going in the data club data. And this is the function of osteocalcin, which is to increase testosterone synthesis and therefore to increase all secondary sexual characteristics. In absence of osteocalcin, mice have small testes compared to wild-type littermates. But in absence of ESP, that means in a gain of function for osteocalcin, mice have a bigger testis. And so we have used back and forth this internal control in the system for the experiment we have already reported and for the experiment I will present today. So the function that osteocalcin regulates, glucose metabolism as a whole, spermatogenesis, insulin sensitivity, are physiological functions that go awry with aging. They are not the only physiological functions that go bad with aging, but certainly this one go bad. You don't need to be obese when you are past 60 years old to have type 2 diabetes, and male fertility is a function that declines with age. And that has brought now a hypothesis to the lab, which is really the overarching hypothesis of the work we are doing. And this hypothesis is that bone is in fact a schizophrenic tissue. It has two identities. On its day job, it is a victim of aging, and we all know that after menopause there is osteoporosis. We are not going to speak about it today. But there is another identity of bone that can be defined and now can be tested, and that in fact bone is a gatekeeper of youth, or at least delaying aging, by acting in a series of organs that have yet to be defined to maintain the physiological functions that this organ fulfill. This is a hypothesis, but it can be tested because we have the loss and gain of function, and also because we have now a receptor whose expression is rather broad to test this hypothesis. And in the context of this hypothesis, which is now being tested in several organs in the lab, one organ that comes to mind is muscle, because sarcopenia is by essence a manifestation of aging. So I'm not going to labor this point, but we all know since yesterday, since we did not know it before, that sarcopenia is a hallmark of aging, and it often manifests itself simultaneously with bone loss. Its cellular, endocrine, paracrine, molecular underpinnings are not fully understood. I think it's a fair statement. And therefore, we were interested to know if, in fact, osteocalcin was contributing to maintaining muscle mass, and if the absence of osteocalcin or decreasing bone mass would affect, would precipitate sarcopenia. So we looked in osteocalcin null mice, and I think that maybe the first thing to see on this slide is that we looked at 6 months of age. We looked before, as I will show you, but this is a manifestation in part that occurs with aging, and what we see is that whether we look at soleus, tibia anteralis, gastrocnemius, quadriceps, biceps, any muscle, there is a 10 to 20% decrease in muscle mass in the osteocalcin mice that manifests itself around 6 months of age. It does not manifest itself around 6 months of age. It becomes significant around 6 months of age. We looked also at mice that lack what we call OSTAR, because it's easier to pronounce, the osteocalcin receptor, in all cells. So it's not a cell-specific knockout, but the OSTAR null mice have the same sarcopenia than the osteocalcin mice, and it develops at the same age. And we use that as a control the ESP null mice, and in absence of ESP, mice have an increase in muscle mass, not in all muscle, but in the most muscles that were affected by osteocalcin. And again, we did not pursue the analysis in older mice, but I'm sure that in fact in the ESP null mice at older age, there will be an increased muscle mass in all muscle. So clearly, in absence of osteocalcin, there is a decrease in muscle mass, there is sarcopenia. If we look histologically at the muscle of these mice, what we say is that there is a decrease, if my arrow is here, a decrease in the diameter of the fibers in the muscle of osteocalcin null mice. From now on, I'm going to show only the data in osteocalcin null mice, but we looked at the 3 models. The number of fibers is not decreased, but the diameter is decreased. And if we look at the cell diameter, looking at different diameters, one can clearly see that in the osteocalcin null mice, the diameters are smaller, and if we look for bigger fibers, they are absent, or virtually absent, in absence of osteocalcin. There is, in absence of osteocalcin in muscle, a decrease of oxidative capacity that can be measured by many ways. One is, mitochondrial protein content. It's decreased 20%. Mitochondrial protein per muscle weight, and mitochondrial DNA. It's all decreased by at least 1/3. As a result, if we look at this marker of the oxidative function of the mitochondria, it is decreased. As you can see, the succinate dehydrogenase staining is completely gone or virtually gone in absence of osteocalcin. And this can be quantified for 2 markers, succinate dehydrogenase and citrate synthase, that is also a marker of mitochondrial activity. So that's the second aspect that is affected by osteocalcin, the lack or the gain of osteocalcin. And there is a third one, which I don't think is probably the most important one, but again, we don't have all the data, is that in absence of osteocalcin, there is a decrease in the cell division in muscle earlier than in 6 months of age. This is explained because there is a decrease in cyclin D2 expression in absence of osteocalcin. You can also see that there is a decrease in PGC1 alpha expression that explain the oxidative phenotype I presented. There are few other genes that are affected. We are looking at them, and at others we don't have all the data. But we have few data about cyclin D2, which has been shown to be important in heart when heart goes through hypertrophy in another models. So in absence of osteocalcin, there is a decrease in cyclin D2 in biceps, EDL, and in soleus. And if we treat isolated myoblasts with osteocalcin at increasing dose, but as you can see here, the doses are very small, 0.3 nanogram per ml, we increase cyclin D2 expression in myoblasts in culture, whether they are primary myoblasts or cell line derived. If we look at now structural genes, there is a decrease in myosin heavy chain 2, 4, and 7, and there is also a decrease in slow muscle troponin C in osteocalcin mice. I don't show you the data here, but all the expression of all these genes is increased in absence of ESP. So the question is, and we heard yesterday the point being made over and over by Steve Cummings, mass is mass and only mass, it's not function. So what are the functional consequences of this decrease in muscle mass, which is by definition sarcopenia? So we are in the midst of this experiment, but what we did was to put mice on treadmill and to ask 2 questions. Can they run like a sprint, or can they run like a marathon? So we took heterozygote and homozygote mice, and you can see that in absence of osteocalcin, the running distance, the maximal speed, and the time on the treadmill is decreased in the sprint experiment, and we can really quantify it, and it is significant for the homozygote mice. It is not significant for the heterozygote, but almost significant. It is, the phenotype is even more severe if we look for a long distance running. Putting the mice on treadmill at a constant speed, you can see that the distance run is decreased 30% in this experiment, and you will see another one where it is decreased 50%. The time on treadmill is decreased, and if we quantify, the curve is shifted to the left, indicating that in fact the mice cannot run, or at least they don't have the muscle to run as well as wild-type mice. So there is a functional consequence of this decrease in muscle mass. It is, in fact, time-dependent, and this is why and where the aging component of the phenotype appears. If we look at the endurance test, the mice run normally at 6 weeks of age, and they also run more or less normally at 8 weeks of age. However, starting at 12 weeks, we now see the phenotype appearing, and it gets worse over time. And it's consistent with the notion that bone health is here, in fact, to prevent aging in other organs. So this phenotype is GPRC6A mediated. Again, we don't have a cell-specific knockout. But if we look at mice that like GPO star in all cells, what we see is that they have a decrease in running distance in treadmill that mimic, in fact, phenocopy almost completely what we saw in absence of osteocalcin. Again, as I said, what we saw in osteocalcin, we see the mirror image in mice that lack ESP, that have too much osteocalcin. The time on treadmill is increased, the running distance is increased, the maximal speed is increased, and the distance is increased also in the long distance running setting of the machine. This is a function of bone-derived osteocalcin, and we did an osteoblast-specific knockout of osteocalcin to be absolutely sure that there was no cryptic expression of osteocalcin in muscle that could explain this phenotype. And when we do an osteoblast-specific knockout of osteocalcin, what we see is that, in fact, the distance run now is decreased 50%. The total time on treadmill is also decreased more than 30%. And the curve is even more shifted to the left. These mice really cannot perform, cannot walk, cannot run. So in the last aspect of the work, that is again a work ongoing, we ask another functional question. Is osteocalcin needed for muscle regeneration? And to do that, what we did is to inject cardiotoxin in one muscle on one side and use a contralateral muscle of the same mouse as an internal control. So when we inject cardiotoxin and we look if the muscle can regenerate, what we see is that, in fact, the percent of increase of muscle mass after injury is decreased roughly 30% in all muscle testes, tibia anterior, gastrocnemius, quadriceps, in the absence of osteocalcin. And that results in a decrease in muscle weight that, in fact, is never compensated after the injury. This regeneration, in fact, is not an early or late event. It occurs immediately after the injury by cardiotoxin, and the mice, in fact, never increase, as you can see, their percent increased muscle mass does not change whether we look at 12 days post-injury or at 90 days post-injury. It's the same. They don't move. They cannot regenerate. One reason for that, and I don't think it is the only one, but that's the only one we have for now, is that there is a decrease in muscle stem cells in absence of osteocalcin mice. The PAC7 satellite cells are decreased after cardiotoxin injection. And this does not increase over time. So the mice don't have the pool to regenerate as they should after injury. There is a decreased proliferation in osteocalcin muscle mice following regeneration, and we are now working more to know what are the genes beyond and beside cyclin D2 that can explain this phenotype that is, in fact, quite striking. There is no apoptosis defect in these mice. The reason why osteocalcin is needed for regeneration is that if we inject cardiotoxin in muscle of wild-type mice, what we see is a twofold increase in OSTAR expression in muscle. So OSTAR expression is regulated more than osteocalcin is, and this explains why this is one of the explanations why osteocalcin is needed for muscle regeneration following injury. And not surprisingly, if we look at the response to injury in the OSTAR null mice, we can see that in wild type, cyclin D1, PAX7, MyoD are increased following injury. If we look at now OSTAR null mice before and after injury, cyclin D1, PAX7, or MyoD are really not affected. So, in summary, it seems that among the function of osteocalcin, the regulation of muscle mass and muscle function is one of them, and it's again a function that goes bad in aging, and to be more precise, bone regulates muscle mass, muscle function, and muscle regeneration. It does it through osteocalcin, which promotes myoblast proliferation, and we have reason to believe that it promotes other aspects of myoblast molecular function. The molecular basis of this function are not known yet, but if we look beyond muscle, and I know it's a bone and skeletal muscle meeting, but still, if we look beyond muscle, the existence of this function of osteocalcin supports the hypothesis that healthy bone delays aging in multiple organs. So this is the work of a single postdoc in the lab, Catherine Lowe, and I have to thank the NIA, the NIDDK, and the NIAMS to pay the bill. Thank you very much.
Did you turn on the microphone?
What is the time course or manifestation of the other physiological effects of loss of osteocalcin that you nicely described with respect to then the development of the muscle phenotype, and how do you control for that?
Yeah, that's a very good question. I forgot to say something very important. All the experiments were done in female mice to avoid the confounding factor of the low testosterone. So that's taken aside. The manifestation of glucose metabolism appears around 3 months of age, more or less at the same time. I don't think that increasing insulin decreases muscle mass, but to address this problem. But it's all done in female mice. I should have precised that.
Yeah, so I have a question. So you, in terms of your functional output, you had treadmill running and that kind of thing, and you know, muscle mass clearly doesn't track with running performance. I mean, if we look, I mean, unless you're talking about sprint runners. And so I was just wondering whether the osteopon could have some effect on the heart. I mean, so I would say your changes in metabolic profile are consistent with metabolic responses to the running. But could there be some effects on the heart or some aspect of cardiovascular regulation in terms of runs to exhaustion?
So we have not seen any effect on the heart. Okay. We have looked and there is none.
So in terms of structure?
In terms of structure.
Okay.
Very nice. This is probably a difficult question.
Then I don't know.
No, but it's an important question. But it's an important one to ask. In the knockout mice, they move less, right?
In the female, no.
They don't move less.
No, this is done in female mice, so they have normal testosterone, so there is not a problem.
So they don't move less, but they have a lower oxidative capacity.
Yeah.
So what's the mechanism, do you think, for the lower oxidative capacity?
Yeah, I was trying to say many times during my talk, we are working on the mechanism.
And that's, so can you speculate?
If I could speculate, I will do the experiment.
All right.
Okay, I have a question regarding the reduction in PAX7 null cells. It looked like it was maybe 20%.
Reduction of what?
PAX7
yes.
stem cells. It looked like it was only maybe 20%. And the ablation studies that have been done suggest that you need to remove the majority of the population actually to affect regeneration. So your regeneration defects look quite a bit more serious than could be accounted for by a minor reduction of cells. So can you speculate on that as well?
No, I don't speculate, never, because I don't think. We just do the experiment. We are now doing cell-specific knockout of OSTAR, and we will see what is the phenotype. I don't think that it is only a satellite cell phenotype, and I don't think either that it is not at all a satellite cell phenotype.
And since these are data that you haven't seen before, I had one other question. Are there slides in there as well of the osteocalcin nulls bred with dystrophic mice to see if the phenotype's worse?
No, we have not done it yet, but we are planning to do it.
Yeah, beautiful work, Gerard. I'm wondering if you can tell me what, so in your animal models, you really have a chronic developmental defect because the gene is missing from birth or from the embryo stage up to the time you test. But if you were to delete osteocalcin in an acute situation with some kind of an inducible Cree deletion event, what would the effect be? Have you thought about that? Would that be different than the chronic effect?
Yeah, I thought about it. So, as you know, there is no osteocalcin expressed in the embryo before E17.5. Right. So, the developmental identity of osteocalcin is a pale one. This being said, we are doing a cell-specific and time-specific knockout of osteocalcin. And in fact, the mice I presented, the osteoblast-specific knockout, are inducible.
Okay.
Very nice, Gerard. In terms of communication between muscle and bone, do we know anything about the concentration of the osteocalcin in the serum of these animals, as is, in other words, how does that osteocalcin communicate or get to from one place to the other? And second of all, have you or are you considering doing studies where you actually add back under carboxylated OC and see whether or not that had an effect independently?
So for the second question, yes, we are treating mice with osteocalcin, but we have already done it because the ESP mice have more osteocalcin. But we are anyway doing it. The first question, we have to assume that osteocalcin goes in blood and then in muscle. I'm not sure I understand the question.
No, I guess that's the thought. So I was just wondering if you had confirmatory evidence of correlative levels going in the right directions up and down relative to your biology.
I don't know how we can address that. We can only address it by looking at the knockout of the receptor in a cell-specific manner and doing double hit with osteocalcin, which is ongoing.
I guess, how about in the ESP animals? Would you be able to do it there?
Well, they have more osteocalcin, we know that.
Final question?
Does the, have you considered the possibility that there's a novel receptor for osteocalcin on satellite cells?
Yes. I think there is more than one receptor for osteocalcin. Whether it is in muscle or in other tissues need to be determined. And again, we are doing the cell-specific knockout, and I think this is the only way to address it, and we have to do it in the two populations of cells. But we don't have the answer for that. If I was to bet, or to speculate, I would speculate that the other receptor for osteocalcin is in another organ.
And one other thing, given the role in glucose homeostasis, does the osteocalcin knockout muscle transport less glucose per gram wet weight than the wild type?
That's something we are looking at now.